Hello students, I am Ms. Triveni Tari from PSS College of Arts and Science. We are going to study a module from Unit 4, Bryophytes, and the module is Reproduction in Riccia. Outline includes reproduction in Riccia, that is vegetative propagation and the sexual reproduction. Learning outcomes: enlist the modes of vegetative propagation in Riccia, describes the modes of vegetative propagation in Riccia, explains the sexual reproduction in Riccia. Now let us discuss in detail the reproduction in Riccia, wherein we have got the two major modes. The first one is the vegetative propagation and the second one is the sexual reproduction. Let us go into the details of vegetative propagation first. We have the four modes. The first one is the fragmentation. The second one is the adventitious branches. The third one is the persistent apices. And the fourth is the tuber formation. The first one, that is fragmentation. Here we see that the older portions will die and the lobes will separate. In the picture you can see the thalli lobes which are growing. With the due course of time, the older portions are going to get decayed and the remaining fragments are going to separate and form the new plant. That is called as the fragmentation. The second one is the adventitious branches. From the ventral surface of the thallus, special adventitious branches develop. As you can see in the picture, the small bird-like structures will be arising, which are called as the adventitious birds. They will later get detached from the parent due to the decay of the connective tissue and develops into a new plant. So that is the adventitious branches. The third one is the persistent apices. The thallus lobes are killed due to prolonged dry conditions except the apical portion. As you can see in the picture, the darkly green colored structure that you can see, those are the persistent apices. These apices will survive deep into the soil and in the subsequent rainy season resume growth and form the new plant. The fourth one is the tuber formation. The apices of the thallus become thickened to form tuber at the end of the growing season. Under the unfavorable conditions, plant perish and the tuber remain dormant in the soil, which resume growth under the suitable conditions. So as you can see in the picture, those are the tubers which are formed. Now let us study in detail the sexual reproduction. Here the gametophytes are involved in the production of the gametes. The species of Riccia, they can be either monoecious or they can be dioecious. The male gametophytes are referred to as the antheridia. So they will be having the male sex organs. And the female gametophytes will be bearing the archegonia, that is the female sex organs. Now here the sexual reproduction we are going to discuss with respect to the distribution of the sex organs, the structure of the sex organs and the fertilization. Let us study in detail the distribution first. The sex organ, that is the antheridia and the archegonia, are developed on the thallus. Generally lie in the dorsal furrow or groove sunk deeply, each in a separate cavity. These are found developed in acropetal order, that is, the younger ones will be near the growing point and the older will be away from it. Now let us study the structure. First we'll discuss the structure of antheridia. Antheridia is an elongated structure. It consists of an ovoid or a pear-shaped body seated on a short fused-celled stalk. As you can see in the picture that is showing the structure of antheridia, you have the antheridial chamber in which the antheridia will be located, that is attached to the thallus by means of a multicellular stalk. And the antheridial chamber is opening to the external by means of an opening called the ostiole. The antheridia will be having the presence of the antheridial wall which encloses the androcyte mother cells. And these androcyte mother cells will get developed into the sperm, that is elongated and having the biflagella. Then next we are going to discuss the structure of archegonia. that is the female reproductive structure. It is a flask-shaped organ and consists of two main parts. A swollen portion called the venter and a long slender neck. It shows the presence of a distinct jacket layer. In the picture, we can see the structure of the archegonia. As we have already seen, it will be basically consisting of two parts. The venter, that will be having the venter canal cell and the egg cell in the venter cavity, and it will be lined by the venter wall. And the upper slender neck, that will be enclosing the neck canal cells, and at the apex, it will be having the presence of the cover cells. So this is the basic structure of the archegonia. Then the next is the fertilization process. Now once the egg and the sperm matures, the next step is the fertilization. The mature male gamete reach the female gamete for fertilization with the use of the water current. So in the picture, it is showing the structure of the mature archegonia, where we can see that the cover cells have opened due to the absorption of the water, and there is dissolution of the neck canal cells as well as the venter canal cell that has made the passage for the entry of the sperm. So the sperms are going to reach the egg cell, and further the male nucleus is going to get fused with the female egg nucleus. That marks the formation of the zygote, and the formation of zygote marks the beginning of the sporophytic phase. So in the sporophytic phase, we have the three sub-stages: the zygote, the embryo and the sporogonium. So the zygote is the first phase where it is nothing but the fusion of the male gamete with the female gamete. Then the zygote gets transformed or developed into embryo. So repeated division and cell enlargement will be seen over here. And the third is the sporogonium, which undergoes sporogenesis to form the spore. Now let us discuss the structure of the sporophyte. Now here the most important point is, in Riccia, the sporophyte lacks foot and seta. Only spore sac or capsule is present. In the picture, we can see that there is outer layer of calyptra that encloses the meiospores. Now, initially, the capsule wall will be enclosing the spore mother cell that undergoes meiosis to form the meiospores. Now, here there are no elaters which are present. So, this is a simple structure that is seen for the sporophyte of Riccia. Now let us study in short the life cycle of Riccia. We have the Riccia thallus, that is the gametophyte, that is haploid. Now whether it is monoecious or dioecious, based on that you will have the antheridia and archegonia. They may be present on the same thallus or they may be present on the different thallus. So you have the antheridia that shows the presence of the sperm, and the archegonia that shows the presence of the egg. So once sperm and the egg matures, then the fertilization will result into the formation of a diploid zygote. The zygote will show repeated division to form the embryo, that is also diploid. Embryo will get transformed into sporogonium, that is nothing but the sporophyte, that is also diploid. Then it will differentiate to form the spore mother cell, which undergoes the meiotic division, and then you have the formation of meiospores, which are haploid. These meiospores are again going to germinate to form the gametophyte, that is haploid. So we get to see the alternation of generations, and the type of life cycle that is exhibited is haplodiplobionic type of life cycle. So this is the schematic representation of the life cycle. These are some of the online and the offline references for your further reading. Thank you.